Okay, it is a great honor for me to introduce our next keynote speaker, Dr. Kafui Dzirasa from Duke University School of Medicine in the United States. Dr. Dzirasa completed a PhD in neurobiology at Duke University. His research interests focus on understanding how changes in the brain produce neurological and mental illness, and his graduate work has led to several distinctions, including the Somian Award for Most Outstanding Dissertation Thesis, the Ruth K. Broad Biomedical Research Fellowship, the UNCF Merck Graduate Science Research Fellowship, and the Wakeman Fellowship. Dr. Dzirasa obtained an MD from the Duke University School of Medicine in 2009, and he completed residency training in general psychiatry in 2016. Dr. Dzirasa received the Charles Johnson Leadership Award in 2007, and he was recognized as one of Ebony Magazine's 30 Young Leaders of the Future in February 2008. He has also been awarded the International Mental Health Research Organization Rising Star Award, the Sidney Prize for Schizophrenia Research, and his laboratory was featured on CBS 60 Minutes in 2011. In 2016, he was awarded the inaugural Duke Medical Alumni Emerging Leader Award and the Presidential Early Career Award for Scientists and Engineers, which is the nation's highest award for scientists and engineers in the earliest stages of their independent research careers. In 2017, he was recognized as 40 under 40 alumni, 40 under 40 in health by the National Minority Quality Forum and the Engineering Alumni of the Year from UMBC. He was inducted into the American Society for Clinical Investigation in 2019 and the National Academy of Medicine in 2021. He has served as an associate scientific advisor for the Journal of Science Translational Medicine, a member of the congressional mandated Next Generation Research Initiative, and on the editorial advisory board for TEDMed. He currently serves on the advisory committee to the NIH director. Dr. Dzirasa is an investigator of the Howard Hughes Medical Institute and an associate professor at Duke University with appointments in the departments of psychiatry and behavioral sciences, neurobiology, biomedical engineering, and neurosurgery. His goal is to combine his research, medical training, and community experience to improve outcomes for diverse communities suffering from neurological and psychiatric illnesses. We are very happy to have you with us. Join me, welcome Dr. Dzirasa. The floor is yours.
Well, thank you to the organizing committee, and thank you all so much for having me here. Anytime someone reads the list of awards, I'm reminded that I spent way too much time in school. So I'm a psychiatrist and a neuroscientist and an engineer. And I'm going to try to walk you through how we've spent years now trying to fuse these disciplines and schools of thought together to solve the challenges that I ran into in the clinic. And this is going to be my first time presenting this data. This is sort of a disclaimer, right? What this means is that at the end, I'll be hoping to get your feedback on how to improve the work. The communication will be broad and we'll focus on a lot of concepts. I am not of the impression that everybody has done training in psychiatry, nor that everyone is in neurosciences, nor that everyone has done machine learning. So I'll try to keep the concepts broad so that everyone can grab onto them, and then we'll sort of dig into the meat of the presentation. And I'll try to do all of that in about 40 minutes to leave some time for questions. All right, so as it's well known, the greatest burden of illness worldwide is due to psychiatric disorders. And this was in 2017. These are things like depression and anxiety, bipolar disorder, addiction, autism. And all of us have experienced in the last few years firsthand the impact of these illnesses on well-being. We've seen this in our colleagues. We've seen this in our children as well. And for years, I used to really think through this challenge of how to define these illnesses in the brain. And if we can come up with an understanding of how these emotions are encoded in the brain and how they become dysfunctional diseases, perhaps we can ultimately come up with treatments. Now, it turns out, as you're thinking about psychiatric disorders, they ultimately come down to two things. And that is social dysfunction or occupational dysfunction, right? So you have to have one of those two things. And the example I'd like to use is like, let's say a patient comes up and says there's a squirrel in their head, right? One might ask, can you see the squirrel in the head? One might ask why they believe they have a squirrel in the head. But what a psychiatrist is really concerned about is, given that you believe there's a squirrel in your head, how does it disrupt your social function or your occupational function? And that's what's really core for how we think about these things. But I don't want to get rid of my curiosity in neuroscience, which is seeing the squirrel, nor the patient's belief about it. So I got really interested in this question about social behavior, because you would constantly inquire or run into people with challenges in social behavior in the psychiatric setting, right? So whether it was a patient, we would see in the emergency room, and sometimes their social behavior would be extremely off. We'd be concerned about them, and then we give them treatment to sort of diminish the disruptive social behavior they were demonstrating in that state. So maybe they were flipping over a table, or as I saw people in the ICU, older individuals, the sun would go down, they start ripping out their cords, right? This disruption in social behavior. And it really made me think about this question of aggression, right? Like aggression towards self and aggression towards others, and what that looks like in the context of social behavior. How does the brain encode it? And could we sort of use that rule, that language of how the brain encodes aggression to figure out ways how to turn off aggression without turning off everything else, right? So when we gave people medication in the emergency room, like it got rid of the aggression, but then it also sort of got rid of their ability to tell you about the squirrel in their head, right? And if you think about this in terms of maybe grandma ripping the cords out, the last thing you want grandma to do is no longer to hug the grandchildren, right? Or in the case of autism, perhaps you want to figure out how to give the child more behavioral regulation, but you don't want to turn off its social function altogether. So I started wondering about this question: is it possible to understand how social behavior is encoded in the brain? So how does the brain deal with different types of social behavior? How does it select them? And then can we sort of turn one of them off and leave the other ones intact? So throughout the talk, I'm going to give you the simple assay that we're going to use. We do this in mice. I'm going to show you this assay, but I'm going to put this up over and over and over during the talk. So we're going to do a simple assay. We're interested in a strain of mouse called CD1s. They're a little bit bigger. And we tend to select this mouse because they're retired breeders. They're a little bit bigger. They're a little bit more aggressive. And the idea is, if you expose this mouse and you put a smaller mouse in its cage, the mouse will attack it. So it'll show aggressive behavior. If you instead of putting a male mouse in its cage, you put a female mouse, it doesn't attack the female mouse. It might sniff it, investigate it, might engage in sexual behavior. And if you take, in the third case, a castrated male mouse, it won't attack it at all. It just sort of exhibit friendly behavior towards it. So you essentially have this paradigm where the same mouse, you can induce aggression, you can induce friendly male behavior, or you can induce sexual behavior towards females. And so we're going to use this paradigm throughout the talk to try to understand, how is the brain selecting these behaviors out of the same animal, out of the same brain? How's it doing this selection as with these exposures? All right, and I'm going to anchor this. Again, I'll sort of jump back and forth from neuroscience to machine learning to stuff I observed in the clinic and why this runs into a problem. So I'm going to orient you pharmacologically. This is sort of the best treatments we have available in the clinic. This is an example of an alpha-2 agonist. It's basically a medication that we use to sort of decrease in young children oppositional behavior. In other words, like aggressive behavior. And I'm going to show you what that looks like in our mouse context, in our paradigms. So we can put our subject mouse in with the normal mouse, and it will attack it. So that's what you see. We give it saline. And then we give it this drug, and you can see what this does. In this case, the drug reduces aggressive behavior, right? So it's a useful drug. The problem is that it also then decreases social behavior, right? So it decreases its pro-social behavior towards the females and the castrated males. So this is sort of highlighting that the problem that I defined that we saw in the clinic, you can also see this problem with the medication in the animals. In other words, it suppresses aggression behavior, but it does that by getting rid of the whole social code. All right. This is an example of some human work. On the left, I'm showing you a study that was done using transcranial magnetic stimulation. The idea is you could sort of control magnetic fields to have local electrical stimulation in the brain. So it's non-invasive brain stimulation. And they're actually doing this on prisoners and, in fact, murderers. And they are stimulating their brains and then monitoring how angry they are and how aggressive they feel. So you're getting now a subjective assessment from humans about what this stimulation, how it makes it feel. And they have a sham group as well. And the summary of this is that by stimulating frontal cortex, in this case, the prisoners express less anger, right? And less aggressive feelings. So this sort of stimulation of frontal cortex could decrease this perception, the subjective experience of aggression. On the right, I'm showing you a mouse study, right? And they're using optogenetics in this case. The tool, what you simply do, imagine how your eye works. It takes light and changes it into chemical energy. It's essentially an ion channel that takes light and changes it into chemical energy or electrical energy. So light hits a channel, channel opens up, ions go in, ions are electricity. So light to electricity. You can express this on specific brain cells. So blue light hits the brain cells fire. So they're doing a stimulation in the part of the mouse's brain in the frontal cortex, and they're measuring the aggressive behavior in the mouse. And this stimulation also decreases aggressive behavior. So both in this human TMS experiment and in the mouse experiment, you can stimulate this area and decrease aggressive behavior. So we wanted to see what this would look like in our hands. We did an experiment. We put this channel rhodopsin in the frontal cortex, and then we're just going to stimulate animals during our paradigms, right? So with the 3 exposures, we're either going to put a female mouse in the cage, a castrated male, or an intact male. The way channel rhodopsin work is blue light makes the channel sort of open up. So blue light, cells firing. The negative control is yellow light, which does not cause the channel to open up. So we're going to compare blue light to yellow light. And then we just do a standard stimulation, 20 hertz, so a fixed frequency stimulation. And I'm showing you in the yellow case, the animals show aggressive behavior towards intact males. And when you look at the blue light condition, this stimulation of this area, as we would expect, decreases the aggressive behavior. But when we look at the other 2 conditions, it looks like it actually decreases social behavior in that condition as well. So the first, looking at an aggressive context, that was a published work that we basically repeated. And it turns out the reason this works is because you're actually getting rid of the whole social code, right? So a lot of times when we think about states and causal manipulations, we manipulate a brain cell or brain area and we infer what that does. But it turns out what it's doing here is it is involved in social behavior more grossly. All right. So then the question is, how does the brain tell the difference between different social behaviors? Or if I translate this into millennial speak, where is the code? And so the talk is about our efforts to try to find this code. So what we've classically done in the lab is we record electrical information from many sites in the brain simultaneously. We're getting both the firing of action potentials, so single cells firing, but we're also getting local field potentials. And local field potentials, like EEGs, we're just getting that from inside of the brain, from the tips of wires, and it's giving us essentially population level activity of what's going on. Whether these codes, these LFPs or local field potentials are primary information or they reflect single cell firing, that's another talk. What we'll say is at a minimum, we just need it to reflect cellular firing, and there's not lots of evidence to suggest that it does. So we can record this electrical information, basically brain waves from many sites of the brain at the same time. And then we can do standard engineering things like filter them across frequencies, ask how much activity there is in different frequency bands of these oscillations. So when we were starting off, we asked what areas to target. I'll tell you a little bit about the scientist who led this work at the end. But she went through and she came up with a set of criteria to decide which area should we be looking for this code. So she went through the literature, and an area had to be in 3 papers in order for us to decide to target it. It had to be in a study where someone had recorded that brain area and showed that there's information in that brain area that reflected the behavior. In other words, it was an observational study. This could either be through recording cellular firing, or it could be through looking at CFOS, which is cells that become more active show an expression of this molecule, right? So you can basically expose animals to aggression, sacrifice them, look at this brain. There's more CFOS in the brain areas that are activated in aggression. Secondly, she wanted to see a manipulation study. In other words, someone either put an electrical wire there, stimulated, or optogenetically, and it changed the behavior. And so she had to find at least both of those types of papers in the 3. And then that brain area had to be mentioned in a big review. That area had to be mentioned in a big review of aggression, right? So when we're doing things, we want to make sure that the things we choose are anchored in the broader literature, because then people are more likely to accept whatever we find. She picked areas also that were translationally relevant. So we know there are things like olfactory bulb that are mice are really involved in aggression. It's unclear how much humans use the same area in the same way. And so we didn't include those in our choices. And we avoided areas that were like difficult because of our, we had to implant the animal. So things that were more technically difficult to access, like periaqueductal gray, we didn't include. And then we included two areas that were, one that was interconnected with all the brain areas we chose, medial-dorsal thalamus, and then what is our negative control area, which we learned a lot about negative control areas in the brain as a result of doing this, primary visual cortex, all right? So the areas that we implanted are listed here in the middle: orbital frontal cortex, infralimbic cortex, prelimbic cortex, nucleus accumbens, medial amygdala, lateral habenula, medial dorsal thalamus, ventral medial hypothalamus, ventral hippocampus, and primary motor cortex. And what we're going to do experimentally is we're going to implant a bunch of animals, and then we're just going to expose it to the three conditions, right? So we'll put the female in, take the female out, put the castrated male in, take the castrated male out, put the intact male in, put the intact male out. And then we're going to do that same series of exposures over a series of days. So every week we come back and we do this again. And this allows us to, over a series of experiments, record hours of electrical information from each animal during the course of these experiments, right? So we're able to gather a whole host of data of how the brain is responding to each of these exposures. And another reason why the LFPs are really important is if I put a wire in a brain and I record, 5 cells in an animal, and I go to the next animal and record 5 cells, I'm not really sure how those 5 cells relate to each other. Because the LFPs sampled at a higher level of analysis, I can infer whether, to some extent, which that signal generalizes, right? It's the same sort of thing. If you have chest pain, you go to the doctor, they put an EKG on. We assume that that signal generalizes from person to person, even though if I was to record individual cells in your heart, I wouldn't be sure how those mapped in the same direct way. All right. And then this is an example of the power spectrograms. It's just that oscillatory signal that I showed in the LFPs. We've deconstructed across frequencies. The colors, the more red it is, the more activity there is in that frequency. And I'm just showing you here non-aggression. So when it's engaged in social behavior on the left, non-aggressive social behavior, and aggression on the right. And I'm not going to ask you to extract anything from that image, rather than just look at it and say, maybe there's a little more yellow in the one on the right, right? So what we're going to do experimentally is we're going to take all of those seconds of time when it is aggressive, all of those seconds of time when it's engaged in social behavior with the female and with the castrated male. And we're going to take that data and essentially try to learn what is different with like a simple decoder, right? So can we take lots of brain information, figure out how to distinguish aggression from the other two, so learn what that state looks like, and then see if we can go to a new animal that we've never seen before and decide whether it's being aggressive or whether it's engaged with a female or whether it's engaged with a castrated male. So here's the setup. We're going to record electrical information from 20 animals over a series of sessions as exposed to those three conditions. We're going to gather all of that data. We're going to take all of those seconds, pool them together, and learn each brain area, right? What's the pattern for aggression? What's the pattern for social behavior with the female and the castrated male? And then we're going to go to 9 new animals. And in those 9 new animals, we're going to see if we can figure out what each and every single one of them are doing, right? So we're not now trying to decode seconds. We're trying to decode the animal state that new animal is in. And so here's what that looks like. First, and I'm going to walk you through what we're trying to decode in each situation. We take all the data when the animal's in isolation, and then we're going to try to decode whether the animals are engaged in social behavior at all, right? And we're going to do these for each of the brain areas. On the data, I'm showing you the plots. 0 basically means no information. 0.5 is perfect decoding. It's basically an AUC of 1. And I'm showing you each individual brain area. The bar graphs are showing there the mean and the standard area for those 9 animals. So in other words, the 9 animals, each of them individually you can decode. And so look at all the brain areas. All of the brain areas have a social code in it, right? And so with each of these brain areas, you can distinguish whether the animal is in isolation or if it's socializing. So that's condition 1. We know there's a social code in all of these brain areas. Secondly, we look at aggression versus the prosocial states, right? So same thing, we're going to take those 20 animals, we're going to learn what the pattern is for aggression versus the other states, and then we're going to go to 9 new animals and see if we could tell if those 9 new animals are being aggressive or in a prosocial state. How good is the code? And there are 4 brain areas that come out as being useful that separate aggression out from the other 2 states: medial dorsal thalamus, ventral medial hypothalamus, ventral hippocampus, and primary motor cortex. And so those look like they have an aggressive code. Then we go to the castrated male versus the other 2. None of the brain areas can separate out the castrated male from the other 2 states. And so there's no codes there that do it, particularly the brain areas that we're looking at. And then finally, we go to female versus the other 2 states, and their lateral habenula is particularly useful. All right. So we basically have gone through all of this, looking at each individual brain area, and we were super excited at this point in time because we'd found 4 codes, right? Lateral habenula, medial dorsal thalamus, ventral hippocampus, and primary visual cortex. Interestingly enough, primary visual cortex was our negative control area. Neuroscience has an interesting sense of humor. But we started to think about this some more, wondering what we were actually capturing. So as you can imagine, when it starts being aggressive towards the C50, the poor, happy, friendly mouse, the whole visual scene starts changing, right? So perhaps what we were capturing in visual cortex was just that the visual scene is different when you're being aggressive. And so we started wondering if there were ways to control for all of the other things that may be accompanying the aggressive behavior to see if we can dissect where this code actually was. And so we took those animals and we put them through, those 9 animals, we put them through another paradigm. So we're not learning a new code. We're taking the original code we learned, and we're going to see if it can work in a new aggressive paradigm. And in that paradigm, we take an animal and we put it with an object, and the animal sort of sniffs the object. It hangs around, but the object isn't moving, so the visual scene isn't changing in the same way. And then we take the object out, essentially cover it in male urine from another CD1 mouse, and then put it back in the cage. And when you put the animal, when you put the block in there covered in male urine, all of a sudden the mouse attacks it. So it gets really aggressive towards the block as if it's another mouse. And then we're just going to ask, can these 4 brain areas decode whether you're with a block covered in urine or a block not covered in urine? And when we do this, only ventral hippocampus survives. So the code falls out everywhere else. It's only ventral hippocampus that's tracking this new behavioral code. So we're really excited here, but we wanted to ask a question: if ultimately we're trying to develop some sort of therapeutic strategy for treating humans, we wanted to ask, is this the best code, right? So as I mentioned, we're recording local field potential activity for many brain areas at the same time. So instead of just thinking about what's going on within a brain area, we can actually start to ask cool questions like you all saw with the functional magnetic resonance imaging studies about how brain areas are essentially coordinating together. This says, is there code that's distributed across brain areas in an integrated way? So we can take advantage of engineering principles, and these principles say things that change together across time tend to lie within the same system. So we can quantify this feature called synchrony, and it's just basically extracting how things change together across time. So I'm showing you 2 oscillations here, and you can see that their peaks tend to align. You could see that in red, right? And so we can quantify that. You either hear the term synchrony or coherence. We can also take advantage of tools that are based in statistical forecasting to say, can you predict future activity in one area based on current activity in the other? And if current activity in one area predicts future activity in another, we can infer that information is moving from one direction to the other. So we can quantify directionality. Similarly, we can ask if past activity is related to current activity and further information is moving in another direction. So as you can see, this problem becomes really complicated, right? And suddenly we're recording 11 to 13 brain areas. You not only got information across frequencies ranging from 1 to 70 hertz, you now have brain areas interacting with each other across these frequencies and information leading and lagging. And so a few years ago, we started running into massive problems with our data and figuring out how to make sense of how the brain is having a code when there are that many things going on. And now we're recording many more predictors or many more variables than we are mice. And here's the solution that we came up with, and I'll walk you through this. We did this work with Larry Karen, who's an electrical engineer, also has dabbled in neuroscience and geothermal imaging over the years, and David Carlson, who was his grad student at the time. And to anchor you into this model, I'm going to use the analogy of music, right? So these local field potentials are essentially waves across time. When you hear sound, you're hearing oscillations or sound mechanical vibrations in the air on the order of tens of thousands of times per second. So the best way to think about this model is just to think about music, when the LFPs are the music. And what we're going to do is we're going to take that music and we're going to chunk it up
into those features that I mentioned: power, so amplitude across frequencies, coherence or synchrony, and then what's leading and lagging with Granger. And that's essentially notes. So we take the music and we just chunk it up into notes. And then we're going to use a supervised autoencoder. It's a form of machine learning. And we're essentially going to learn how those notes, in other words, right, when you hear pitch, it's really fast oscillations, how those fast signals change together over a slower time scale. So in other words, what are the notes playing together, or what are the chords? So we learn the chords and the instruments in the music. Then we figure out what are the chords and the instruments and how that relate to behavior through a process called supervision. And then finally, we build one last feature on this model that I think is critically important, and it's that we ensure that it generalizes. And by generalize, it's if you hear music with a new set of instruments or at a new tempo, you still know it's the same music. And this is really important if ever you're going to think about a clinical therapeutic. You want to make sure it works on the next patient that comes in the door, right? And so we have a feature built in here that both decodes the networks and then ensures that it can reconstruct the original data, and that ensures generalization, right? So this is how it's built. And what we're going to do is we're going to take the exact same approach I just told you. We're going to take those animals that we've recorded. So we've got all the data, right? We got brain information from all the brain areas. And we're going to take that local field potential data and build it into this model. And then we're going to train the model using the 20 animals, the same 20 animals that we use to train the individual area models. And then we're going to ask, can our model decode 9 new animals? So can it tell you what each of those 9 new animals are doing? So when we do this, we get a series of networks out of the model, right? 8 is here. I don't have time to get into why we ultimately ended up with 8, but it explains the variance of brain activity that we observed across the task. And one of them is supervised. So that's the one we sort of have put the most weight on to learn the difference between aggression and the pro-social behaviors. Here's what the performance of that network looks like compared to ventral hippocampus. So I'm just showing you the AUCs here. The network, animal by animal, outperforms the individual area. So there's more information about what is going on, in this case, from an aggression versus pro-social behavior state, looking at the network. Now, there are a couple of really interesting things that popped out of the data. These weren't built into our model. As you probably can see on the left, our network is actually below 0.5, right? So it means it's actually encoding aggression in a sort of negative direction. So when we looked at what was happening across time, I'm showing you a plot on the left. I'm showing you our different behavioral states. So when an animal is showing aggression, when an animal is showing pro-social behavior towards an animal it would otherwise be aggressive to, when it's interacting with the castrated male, so it never shows aggression, and when it's interacting with the female. And I'm showing you the network's activity, so how loud the cord is playing across time, as an animal begins an aggressive interaction and as it ends an aggressive interaction. And the section I'm showing you in gray in the middle is supervised. So don't pay a whole lot of attention to the fact that it's different. The model has decided that would be different. The windows outside of that, we've basically projected that activity into the network that we learned. So it's not supervised, but it's within subject, different time points. And interestingly enough, when we look at it, you can see that when an animal starts getting aggressive, the activity in the network actually turns off. So we thought we were going to find a network that's activity shot up with aggression. It looked like the brain was actually turning something off and releasing aggression. This was coming out of the data. And interestingly enough, as you look through the different types of social encounters, outside of these supervised window, you can actually decode, there's information in there, whether the animal's about to be aggressive or whether it just was, which made a lot of sense for us. If you've ever sort of observed someone who gets a little ramped up and angry, as soon as they stop fighting, their blood pressure doesn't just go back down to normal, right? So there's something resonant in the system. So we can track the activity of these networks over time. Of course, this is really complex math. And when I started getting data like this back years ago, I needed some convincing that it was real. And so we took a simple approach, which was to ask if this was real, it should correlate with things that we all know are real, right? And so we all believe that there's cells in the brain and that they fire. If this is real, it's at least capturing relevant dynamics if it shows activity that's correlated with cell firing in the brain. So we can do this. I'm showing an example of a cell that we've recorded, and that's a histogram in black on the left, and the activity of the network layered directly on top of it. And we can ask, how much does the network couple or correlate with the firing of individual neurons, right? And so if the network is, in theory, at least capturing dynamics, we should have more than a by chance percent or portion of cells in the brain that are coupled to the network. So a couple principles. One, as we looked across the brain, there were cells in like the vast majority of brain areas that were recorded that were coupled into the network, right? So this isn't just one area doing this. Information is distributed across the brain. We found cells that were correlated and cells that were anti-correlated. But the take home here is that the dynamics in the network is capturing cellular firing. All right. This is what the network looks like. I'm just going to show you this because this is a machine learning talk, so I got to show a really complex data slide before I dig in. What we're doing is we're essentially along the diagonal, right? You can see what's going on in each brain area. It's essentially an autocorrelation with itself. At the off diagonals, we have what's going on between brain areas. That's a synchrony. And with the synchrony, we essentially have brain areas that are leading and lagging when they're synchronizing. I'm going to dig into this a little bit more. This is what we get back. We do some tricks so you can visualize it a little bit easier, right? So these are our circles plots. And what you see is essentially all the brain areas that are recorded around the rim of the circle. We have frequencies here labeled from 1 to 55 hertz. And if there's color around the rim of the wheel, essentially if there's color in the tire of the wheel, it means that brain area and that frequency is part of the network. In other words, it's part of the chord that's being played. If there's spokes going through the center of the wheel, it means that those brain areas are synchronizing on a millisecond time scale as part of the chord that's being played. And I've color-coded that in sort of 3 frequencies so you could see it. One that corresponds with the lower frequency theta in blue, that's about 4 to 11 hertz. One that correlates with beta, which is a little bit higher. And then in gray is the remaining frequencies, just so you can see them. Now, when the brain area is synchronized, again, we're also getting out of our model directionality. So we can extract when things synchronize, what is leading and what is lagging, right? So for each of our brain area pairs in the frequencies, again, blue correlating with theta, green a little bit higher frequency, we can see what's leading and what's lagging. It's obviously a lot of complicated information flowing through a circuit. We've just summarized that here in a plot so you can see it, how information is flowing through the network. So this network is essentially being turned down and it releases aggression, right? So we got really curious thinking about ways that we can validate this network and show that it holds up. You know, before we start thinking about translating this anyway, we want to make sure it holds up to a series of animal behavioral experiments. So I'm going to give you the paradigm that we're going to use to do our validations, and this will sort of use throughout the remainder of the talk. So we're going to take a new set of animals in every case. We are going to record electrical activity from that new set of animals. We're going to do some sort of manipulation that's related to aggression. And then we're going to calculate how much of the network we already learned is present in that animal's brain, right? So we're not going to learn a new network every time. We're now going to take that really complicated space, project it down to the latent space, and ask how that network is responding to these aggressive conditions in a new set of animals, all right? And again, here's looking again at that block and the block covered with urine. The network's activity here, just for simplicity, I've normalized it, and by I mean the like sort of royal we, the lab, which is not me, has normalized this to the home cage, so everything will sort of circle around 1, but you can see how the 2 conditions relate to each other. So now looking at the animals interacting with the block or the block covered in urine, you can see that the network tracks this aggressive condition as well. In other words, the network's activity also turns down when the mice start interacting with the block covered with the male urine. All right, so we got super complicated at this point in time because we like trying to do fancy things. And in the background, we are actually neuroscientists. And so we thought about cellular manipulations and can we do bidirectional cellular manipulations that have been widely shown and accepted in the literature? And can we use them to see if 1, they'll affect behavior, and 2, they'll change the activity of the network, right? And so we're going to first start off with a manipulation that increases aggression. Some of our colleagues had done some work looking at a set of cells in one of the brain areas that we're recording, ventral medial hypothalamus. And it was a set of cells expressing a receptor on the surface called the estrogen receptor 1. And what they found that they could do is they could use another set of manipulation tools. What this one does, it's sort of a combination manipulation tool. It's a receptor that goes on a cell and a pharmacological agent. And when they both come together, the cells fire more. They're called designer receptors exclusively activated by designer dreads. So they use a set of viral tools and a mouse line to basically put the dreads in these cells. And then they gave the pharmacological agent. And what they found was that suddenly the male mice would start attacking female mice, right? So they can get animals to now show aggression in a context that they otherwise wouldn't show aggression. So our experiment was quite simple. We're going to take our mice, breed them with this mouse line that allows you to target those specific cells. And then we're going to put the dreads in, basically redoing the same experiment. So we're going to basically try to activate these cells. The mice should show aggression towards females. And our hypothesis is that the network will suddenly like drop down more. It'll go into the exact aggressive state as a result of this manipulation. And I'm just showing you on the right here that when we test these animals just under normal circumstances, so this is a new group of animals on a new strain. I think a lot of times we don't take that step to test mice on a new strain, right? If we can't get mice to go from one strain to another, mouse to humans is a far cry, right? So we're testing on a new mouse strain here, and the network tracks aggression towards an intact male like it should. All right, so when we do this, our mice, when we give them this pharmacological manipulation, it's called CNO, and you compare it to saline. When you give them saline, you have receptor but no drug. So the animals aren't aggressive towards females. When you put receptor and drug together, the animals are aggressive towards females. When we look at what's going on in their brain circuitry, the network starts tracking it. So all of a sudden, the network responds as if this is a male mouse when it's a female mouse. So we get both behavior and the network activity drop off that correlates with the animals going into an aggressive state, right? So now we can activate cells, induce aggression, and get the behavior of interest out of the other side. All right. But we started wondering, could you possibly go the other direction, right? So maybe we can suppress aggression because we want to like close the whole loop, right? So increase, decrease, and see if the whole thing comes together. Now, this was a little bit more of a tricky problem. So we thought about space, right? But we sort of started the talk on this problem, right? Which is we knew we could stimulate a brain area, in this case, frontal cortex, and we knew that it could suppress aggression. The problem is that it also suppresses social behavior, right? And so what if we ultimately see a change in the network and behavior, but it's not because it's suppressing aggression, it's because it's like turning on off all of social behavior, right? So we weren't really sure how to like sort of get rid of aggression behavior without getting rid of social behavior, which is where we started with this whole journey. But then it occurred to us that we might be able to solve this problem by combining space with time. And so what we'd observed in our experiments was that as animals sort of transition in this aggressive state, you saw a drop off of network activity. So what if we could take the space feature of knowing how to shut up aggression, but then coupling it with the time feature, which is network activity uniquely signals aggression when it drops off, right? And what we would need to pull this off is a way of basically extracting the network state in the animal's head in real time, right? And then when it signals that the animal's brain is in an aggressive state, we sort of turn off social behavior, but only at that time, right? So social behavior isn't turned off. The stimulation doesn't happen when you're in a social state. It only happens when you're in an aggressive state. And that might give us a way to sort of establish the causality of the network. Turns out this is a really complicated problem. Our machine learning analysis takes hours to days. And hours to days is just not quick enough to do closed loop stimulation. And so we came up with a strategy to basically have a reduced encoder. The idea here is that it lacks basically all of the interpretation. So it takes all of our data, right? We sort of throw out the Granger, the leading and lagging, because it's too slow to calculate. We just take our power and coherence features, which we can calculate in real time, and we essentially learn something that couples with aggression. It's not explainable. We don't know like what the features and the shape are. It's just all of the data that we can like smush together to mark the aggressive state. It's prediction, but not interpretation. And then we see, can we still predict the aggressive state, and how well does that couple with how well does it perform against our network, a real network? And it performs relatively well, right? Again, the downsides are, as a neuroscientist, like we can't really make sense of what information is behind this. Think of it as like a neural network. It's really hard to understand all the components, the features, and how they're organized. But the prediction is really good and it's fast, right? So we ended up doing that. And that was a great way to go about this closed loop stimulation. And so what we did was we set for each animal, the threshold, we sort of figured out what's the threshold in which we capture, it's not perfect, right? So we can capture 40% of the aggressive windows. So we're going to try to turn off 40% of the aggressive windows. And we'll by chance sort of hit 10% of the prosocial windows, and we'll live with that. That's as good as we could do right now. All right, and this is just showing you how much information or predictive information is captured by our network versus our what we call our reduced encoder. It's our real-time encoder. They perform equally as well across animals. It's not identical in all of them, but as a population, they perform relatively well. So here I'm showing you now what this closed loop stimulator looks like. I'm showing you when we stimulate the animal's brain with blue light, which again activates channel rhodopsin. So blue light is a stimulator, yellow light is a negative control. And I'm going to walk you through a couple of features here. I'm showing you the network activity that we're recording from the animal while we're doing this. And that network activity is event locked. In other words, it's timed to when the stimulator goes off, either blue light or yellow light. So the first feature that's worth paying attention to is basically a second before the stimulation goes off, network activity drops. This is useful because it means the closed loop stimulator is working, right? It is picking up a drop in network activity and causing the brain stimulator to fire, right? So that's cool. The second thing you can observe here is about a second and a half later, the network activity in the blue case like shoots through the roof, right? And this is what you would hope would happen because, again, as I mentioned, the network activity drops when the animal transitions into an aggressive state. So it is indeed doing physiologically to the network what we would hope that it would do physiologically. And then here I'm just showing you that it is selective for the aggressive state. So the stimulator goes off way more in the aggressive state than it does when it's interacted with the friendly castrated males or with the females. So we get the state selectivity, we have the physiology. The question is what's going on behavior-wise. All right, when we look at this closed loop stimulator from a behavioral standpoint, it indeed suppresses aggressive behavior. And so we were, one, really excited about this. There's a bunch of engineering that went into getting this to work. So blue light compared to the yellow light, the animals show decreased aggressive behavior. So we essentially run this assay, blue light, yellow light, yellow light. Fascinating enough, when we tested it both with the castrated males and the females, it didn't show a difference in aggressive behavior. It has a tendency to drop it in castrated males, but interestingly enough, by dropping the aggressive behavior, it also increases prosocial behavior. So the animal across the board shows no differences in prosocial behavior with a decrease in aggressive behavior. So we were like really excited about this. I'm like, write up the paper, let's get it out the door. And then somebody said, wait, not so fast. We have a problem. Maybe what we're doing here is we're just driving a pattern of stimulation, right? Sort of the right rhythm of stimulation. And that's why we're getting the suppression of aggressive behavior, not social behavior. So it's not real time at all. It's just the pattern over time that happens in that case. So we tried to think about what the best control would be in this situation. And we ultimately realized it was what we'll call non-synchronous control. So here what we're going to do is we'll record the brain activity pattern in one animal, right? So this is what sort of the natural stimulation pattern would look like in a closed loop case. And then we're going to take that and then we're going to play it into another animal on another day, right? So now you have the same pattern of stimulation as the closed loop animals got. It's just not timed to their own rhythm, right? So you might stimulate them before they drop off. You might stimulate them when they're not going to drop off, but you never catch the exact point where the brain is transitioning into the aggressive state. So this is going to be our negative control. And when we test that out, we see that there's no differences in aggressive behavior, right? So in this non-synchronous case, we don't actually get any of the changes that we observed with the closed loop case, all right? So we had one final set of questions, which for us was particularly intriguing, which is, well, if like you're hitting aggressive behavior in some of these cases and you're not, like what else is the animal doing when we're doing all these complicated stimulations? So we just went back and compared our open loop case. So this is our original stimulation. Remember the one that decreased social behavior in all the conditions. And we wanted to know like what was going on with behavior in these cases. And it turns out if you do the stimulation and you put an animal in open field, it just starts running around. So it gets hyperactive. So the reason it's showing decreased behavior, social behavior is because you're making the animal hyperactive, right? And when you stimulate the animal in our closed loop case, it doesn't show that same hyperactivity, which is why you get the selectivity in that case. So I'll end on this last slide. This is sort of the model that we're navigating now, right? your brain is able to transition back and forth between states, and some people are more likely to get stuck in one state than another, right? So you're able to transition from from aggression into a pro-social. Then you could sort of become more oriented to the outside world and exploration. You pay attention to people less. You start looking at every object in the room more, particularly when you've had a really good cup of coffee. But you can transition back and forth between aggression and pro-social and hyperactivity. We're unclear if there's a path from hyperactivity back into aggression. We're sort of playing around with that now. The idea is that PFC, or prefrontal stimulation, just sort of pushes you towards the right, right? So it can take you from aggression and push you into hyperactivity. It can take you from pro-social and push you into hyperactivity, but it sort of advances your state towards the right. What we're doing in the closed loop case is we're deciding only to turn on that pushing to the right when you're in aggression, and then we shut it off, right? So we're just sort of nudging the brain out of aggression into a pro-social state that's sort of stabilized there, and then we turn the stimulator off. So that's sort of the concept that we're navigating here. And we're trying to take similar approaches. We're mapping out some other emotional behaviors that I've mentioned. We're looking at affiliative social behavior, we're looking at aggression, reward processing, and our idea is, can we create these maps, these larger scale network structures of emotions in the brain? All right, so this project has over time been funded with a couple cool sources, the Keck Foundation, Hope for Depression Research Foundation, two R1s, another one from the Brain Initiative, and the Howard Hughes Medical Institute. So I'll end on this last slide. Just to make some important points here. I think I've got I'm doing great on time. All right. So most of this work was led by Yael. Yael is here in the room over there. Yeah, wave, Yael, yay. Who joined my lab and didn't have much experience in machine learning at all, or in vivo electrophysiology recordings. And the work is a real testament to when you get in an environment and there are a lot of people around with a lot of different disciplines and expertise, you could do an incredible amount of really awesome work. She partnered with Austin to do this work. Austin's background was math and statistics. He led most of the initial machine learning work, but Yael and Austin got together and just sort of figured out how to talk and solve things. Much of this work being done through the pandemic. So it's pretty impressive. Ali, who's now a grad student at Mount Sinai, was an undergrad in the lab. Catherine is a postdoc in the lab, worked in mouse models of autism, got really interested in figuring out how to map out the networks underlying social behavior, and is applying some of these same concepts to different genetic mouse models of autism. Neil Gallagher was an undergrad in my lab. He then did his graduate work, is now at Cornell. He was the one, as an undergrad, we started working on methods for closed loop stimulation in the brain that ultimately became the state stimulator. Gwenaël Tama graduated, finished her PhD in my lab recently. She's sort of the optogenetics expert that trained all of us, including Yael, on the technology. Scott Russo is on faculty at Mount Sinai. He was actually on Yael's thesis committee when she was a graduate student. And he was the one that did some of that initial early work with the genetic models, looking at prefrontal stimulation and looking at how to make the males aggressive towards female mice. And so what we figured out how to do was basically take the expertise that was in his lab, combine it with the expertise in ours to solve a problem that none of us could have done independently. And then David Carlson was the undergraduate student, or the graduate student that I mentioned, coming up with the original statistical models. He's now on faculty at Duke, and he's the co-PI on all of these grants that I mentioned. And so I think the point I wanted to make there, right, is in these interdisciplinary space, part of the real challenge is to figure out how to get us all into common spaces, looking at the same problems and bring our expertise to solve these challenges that otherwise really doesn't lie in any field, right? Like the brain cares about a lot of things. The thing it doesn't care about are disciplinary boundaries, right? And so we have to figure out how to get past that to solve these problems. And thank you. And I'm hiring and recruiting. So no matter what you do or where you're from, please, 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 we would love to have many of you.
Okay, definitely do it. Great. So we have time for a few questions. Paul.
How do you sort of cancel out just the fact that the behavior is different in the mice? I mean, I understand you're recording from these very specific regions, but what would prevent you from decoding if you had LFPs in various motor regions, given the fact that the mice are doing this? And how how can you be sure that what you're decoding is sort of aggression as opposed to the behavior of aggression?
Yeah, no, so it's a great question, right? So, I mean, for whether it is aggressive behavior or I'm arguing about anxiety or I'm arguing about word processing, the challenge is generally we need animals to behave, right? They don't give us self-reports. And so we will have solved this when we figure out how to do it in humans, right? Part of our underlying idea here is that there are things that are conserved across biology. We can figure out what those substrates are, and then we could start to ask if they extrapolate in higher order species. But we are sort of like stuck with it might be the expression of aggression. We tried to address that by looking at the windows where the animals weren't actually attacking, and there still is a difference there. But it's unclear if we captured something that is totally indifferent to subtle differences in motor output. The other thing, particularly with V1, right? So we sort of thought about this with the urine experiment, right? But I'm not sure there isn't any reason why V1 shouldn't be an aggressive network, right? I mean, certainly the way visual streams might be processed might change whether I'm aggressive or not paying attention as well, right? And so what it means to sort of underlie a code or to underlie the expression of a code, it became this really weird boundary for us to sort through.
But from a scientific standpoint, you don't know, right? I mean, you could do deep lab cut on these mice and say he's being aggressive, go shut him down.
Yeah, so I mean, we can separate these from the behavior mostly, right? But I mean, there are a whole host of other things that are coupled with aggression in this case, right? I mean, it could be that the heart rate is changing or the blood pressure or something else that we're capturing that is so intimately tied to aggression in this case that we would, I mean, our gold standard for most of the things we do is ultimately getting these to humans, right? I think these steps are necessary to figure out how to anchor the science, find substrates that we then ask to generalize. But yeah, it is going to be an underlying limitation of the work.
So it seems like you're focusing mostly on sort of morphological regions in the brain. Have you any knowledge of like a cellular difference and sort of more that kind of thing that's different within the brain regions or even within the same brain region that you might target because you're using, you know, the dreads and optogenetics within a region, I assume specifically. But have you tried to determine cell types or has there been work to do that?
Yeah. So people have done work, particularly, you know, fantastic work, Daylin, David Anderson, Scott Russo in this case, trying to go in each of these areas and see if there are specific cell types that engage in specific behaviors, right? I mean, there is no doubt that there are cellular substrates within regions that play a role in behaviors, but they don't operate independently, right? I mean, I could put the animal to sleep, cause a cell to fire more, and the animal doesn't suddenly start attacking other animals, right? So it is both about what a cell is doing, but also the context in which the cell is doing things that make up the code. We think a lot about, for us about this question is, how do you anchor cell types into a substrate that then extrapolates across animal to animal and species to species? And it might be the case that the cell type that causes this to operate in strain 1 is different than strain 2, but the network level substrate is still the anchoring phenomenon, right? So one of the things we're hoping to do, and we're doing some work now, is integrating our recording approach with something that allows us to do what is microendoscopic calcium imaging. In other words, we can look at the firing of specific cell types and figure out how they anchor into our network, right? So in other words, how do cell types compose our network level substrate? And is that a principle for extrapolating cell types and combining them across animals? But that work is underway. It's also projection types, right? So you could say a cell is defined by its gene expression, or better yet, its proteins, right? But it also could be its projections, right? And so by integrating it with microendoscopic calcium imaging, we can start to build a frame of how structure is turned into electrical dynamics or code.
Lots of work has been done about astrocytes and their involvement. So that's also something else.
Absolutely, no doubt about it. No doubt about it. Yeah.
Thank you for the talk. Methodologically, that was just a tour de force. It was an absolute joy to listen to you not only talk about the science, but the steps along the way that got you there. Really, just congratulations also to Yael. I mean, just really wonderful as a trainee, really wonderful to see how that work unfolded. What was also nice to see was your conceptual clarity around the end goal, which is not only understanding the circuit, but having some kind of translational value. Could you just speak about how you think of bringing this work forward? It was difficult for me to see how that might be clinically applicable, but I'm sure that you've thought through a bunch of that stuff. So
Yes, so that is a different talk that I gave for a year and retired it so I could start giving this talk. And funny enough, this project started a lot before, and the other one was the pie in the sky idea. But where I started my lab was I was interested in finding what these network level substrates are, right? Seeing if they extrapolated across species, because then you get diagnostics for emotions, right? Ways of anchoring them in their biological substrate. But you also get diagnostics for when, you know, the networks for emotions aren't processing correctly, right? You get essentially an EKG for brain states, right? And then the idea was that we could figure out how to stimulate those using closed loop stimulation to basically push you in and out of a state, right? So I started, this used to be a good thing. I started grad school as Elon Musk, right? I was going to essentially create Neuralink and record brain cells, stimulate, and figure out how to create brain pacemakers to change everything. Somewhere along the line, it occurred to me that this would never scale. Right. When you see this type of illness worldwide, even in the first world, there's just too much illness to think about that sort of technology ultimately being a scalable treatment, right? We don't even have that many TMSs in the United States of America. So a couple of years ago, we started working on a different way of modifying the synchrony between brain cells. So could we figure out how to rewire brain cells? And a lot of the technology that we'll be talking about in the lab now is our new tool, which essentially, sorry, it's a whole new talk, but you can read the paper. It's on the preprints online. We're just finishing up now. It basically puts a gap junction between two specific cell types, essentially creates a system that takes electrical information from one into another. So the presynaptic cell becomes your stimulator of your postsynaptic cell. And so we can essentially rewire the whole network level construct and shift it. And we can do this in a way that's long lasting. So that's our idea of how we essentially want to turn this into translational technology, mapping out the whole network and then figuring out where we can push a certain sub part of it to change the whole structure.
Thank you again.
Thank you.
One last question.
I have a question about aggression by female mice. I know that the males are more aggressive, so the natural ones to start with, but I wonder if you think the same mechanisms or the same interventions would have the same effect on antisocial excess aggression by female mice or not.
Yeah, so I don't have an answer. And I have sort of a hypothesis, but better yet, I have an aim one and an aim two. Right? So yeah, Al is currently doing that now. So you can get the female CD1s to show aggression towards a female animal. What you sort of do is you house them with the male, and then you take that male and put it on the female, and then the female will attack the other female. So there are ways of generating female aggression in the mice. We're going to record them and see if the same network level substrate holds up, see if what we learn in the females holds up in the male, and then see if the closed loop stimulation works, right? The data will show whether it's the same or not.
Makes
Yeah.
sense. Yeah, thanks.
Yeah.